Welcome, everyone, to part one of the Can Do MS Drug and Healthcare Access Series on increasing your understanding, making a medicine. Thank you so much to our speakers and our generous sponsors. For more information on the Drug and Healthcare Access Series and other Can Do MS programs, please visit cando-ms.org. Tonight, we have joining us Mike Panzara, who is a doctor and the chief medical officer, and he will be discussing the following topics: developing a better understanding of the drug development process, understanding how an idea for a new medicine gets to a clinical trial, and learning how a new medicine gets from a clinical trial to approval. So with that, I will turn things over to Mike and allow him to introduce himself, and thank you so much for being here.
Thank you, Brian, and it's really great to be here. I do appreciate the opportunity to speak with you all about the drug development process. My name is Mike Panzara. I'm Chief Medical Officer at Wave Life Sciences. We don't work in multiple sclerosis, but previously I was heavily involved in MS. So this is has always been a passion of mine, so it's very exciting to be here to talk to you about the drug development process overall. But I'll mention along the way its applicability to multiple sclerosis. So first of all, really this is a discussion of what the traditional steps are in the development of a medicine. It is a complex process, so I'm going to highlight the major steps. There's a lot of ways, different ways that this drug development process can happen. This is more of the more straightforward approach, but of course along the way I'll also mention areas that there could be some different approaches in the process or some deviations from this. You start from when you select a target, and then I'm going to take you all the way down through the application and the regulatory review process. This slide illustrates simply the overall drug development process, starting from the target validation and selection all the way through to phase 3. And it's these data that are critical in supporting the approval of a medicine in any area. So what do these terms mean? First of all, we're going to start with what target validation is. Target validation and selection. So this is the place where the ideas happen. This is where somebody has an idea that says, I believe that this gene, this protein, this molecule, this compound may be involved in the disease. And I now have to figure out a way, how do I change it in a way that gets it to do what I want it to do? How do I design a drug that gets to the target? How do I show that by my drug getting to that target has the possibility of having a beneficial effect someday? And again, this is where the new ideas come in, and a lot of things fail at this stage, where there's a lot of exploratory work. But you start from where in the pathway, based on what's known about a given disease, can we intervene to hopefully have an effect? And once you have that idea, once you try some work to prove that that may be a relevant idea, you move into the drug discovery phase. So now you have this target along a pathway, and maybe it's a pathway involved in the onset of a disease, or maybe it's a pathway or some part of the body that's involved in the progression of a disease. And now you have that target, and now you're trying to design chemicals, proteins, in my case, oligonucleotides, what we overall call compounds, to try and touch that target, try to deliver the medicine to that target to have the best effect. And we do that through a process what's called optimization. You have could be hundreds of different chemicals or proteins you have a target, now you're trying to see how they influence that target. You look at potency, so how strong is it? Safety, if you hit a target in a certain way, could it lead to unexpected side effects that could hurt someone? So we try and work through that. And then we think about how is this drug, how might this drug be administered someday? How long does it last? Is this going to be a once-a-day pill? Is this going to be a once-a-month infusion? All of that thinking begins here, and you go through a process to hopefully narrow down, as I said, what could be hundreds of compounds, molecules, chemicals, whatever, to a single, what we call candidate. and you go through what, at that point, you go through a stage called candidate selection, and you make your bet. You get to the point where you're going to say, this is the one compound that I'm now going to advance to the more focused development phase of trying to turn this chemical or this protein into a medicine. Now, once you do that, you evolve into a process called preclinical development. This is where you now have this, your chemical, your protein, and you're now trying to understand in cells and in animals, how is this thing going to work? What does it do? We do what's first of all called pharmacology characterization. I mean, what that essentially means is we're trying to see how the drug acts in animals. We say preclinical PK/PD. That means pharmacokinetics and pharmacodynamics. That means how does the drug move through an animal, and how does the animal react to the drug? And then we're trying to do this sort of push-pull, trying to balance, trying to determine what in an animal, and trying to predict what dose levels are likely to be active in humans. And we're trying to test across a broad range of doses to try and give us the widest range to bring to a human, again, to have that desired clinical effect. An important part of not just understanding how the drug how the drug moves through the body is understanding safety effects, and that's called toxicity studies. And these are done typically in a rodent and in a monkey, where we're trying to understand how, when given to animals, how they react, what are the safety signals that we might see. And you typically are looking at single doses, and you evaluate that. You look at repeat doses. If you continue to give the drug, are there new risks? And you're looking at any risks at this point. You're trying to figure out if we give super high doses, if we give low doses, if we give it one way or the other, what are the risks of it that a human might experience sometime down the road when we finally get this to clinical trials? And the goal is to actually characterize that. We have to figure what is the highest dose we can get to, and what does that look like? What does it look like clinically, meaning how do the animals behave when they're given the drug? But we even look microscopically. We look at the organs of the animals, and we try to figure out, are there things microscopically that the animal may look perfectly fine, but then when you look microscopically under the microscope at the organs and the tissues from these animals, you may see microscopic adverse effects that could be a real problem for a human being. So this is generally a pretty robust and intensive process, and we're trying to mimic human doses and mimic what might happen, so we can then, as we're planning our human doses, be able to inform patients, investigators, and physicians about what risks they might see when this is given to humans. Now, all of this activity culminates in what's called an IND application, or an investigational new drug application. This is called a CTA in the US, or in Europe, but the concept is the same. In the US, it's called an IND. Now, you may have heard that term, but what really is an IND? Technically, an IND is permission to legally introduce an investigational drug into interstate commerce. What is interstate commerce? Interstate commerce means any process in the drug, any part of the drug development process, is manufacture, it's packaging, distribution, anything that crosses state lines is subject to an IND. And that could be the ingredients that's in your drug, the packaging that you use to package your drug, and the movement of the actual product across state lines. That is all regulated by the government, by FDA. So why do we need an IND? Because it's the law. The US Code of Federal Regulations basically applies to anything used in a clinical investigation subject to the Food, Drug, and Cosmetic Act, is required to have this permission. And by the regulations, a clinical investigation is any experiment in which the drug is administered to humans. So if there's anything that you want to test in a human, in a clinical investigation where in the United States and outside the US, this is similar rules, you need an IND. And if you don't follow that, if you don't do an IND and you try to do a clinical investigation in human beings, it's a violation of the law, as well it should be. Now, what is in an IND? What is it made of? I talked about some of the different components already, some of that research work, some of that work on understanding the mechanism, understanding how the body treats it, understanding how the toxicity in animals. But there's some core elements that are also in the IND. First of all, the information about, quote, the sponsor. So what is a sponsor? The sponsor is any person or group who is taking responsibility for initiating a clinical investigation. That's often a company that is the sponsor, but it can be an academic person. It can be a physician who has an idea. It can be a patient advocacy group who holds and initiates the IND as a sponsor. The point is that somebody needs to take responsibility for this compound in this plan. And the other important piece of this is how you're now going to advance that compound. And that's what the general investigational plan is. You need to have a rationale and your objectives of where you're going here. What's the clinical plan to get you to a medicine someday? That can involve multiple studies, but you have to have a rationale and justification to do an experiment in a human. So there's a general investigational plan that's going to be an important part of this, so you can tell the regulatory authorities, this is how we're going to make this a medicine. This just isn't something we're just doing without a particular goal in mind. Also, an investigator brochure is part of that. That's what is also known as the label for an investigational drug. When you get a medicine from the pharmacy, I'm sure you've opened it up, or there's this little piece of paper that has all the information about the drug on it. That's called a label. The investigator brochure is the origins of that, is the beginning of the formation of that, where it's the information that we give to the physicians, and the regulatory authorities, to help them understand what we know about the drug at that moment in time. It's something that evolves the more you learn about the drug, but you need that from the beginning to say, based on the studies we've done, this is what we believe, the risks, the potential benefits. That goes in the investigator brochure. You need a study protocol. This is your actual plan for the clinical trial. You have to have a rigorous plan that's designed to answer the scientific question. That could be, is the drug safe? That could be, does it have an effect? You need to have a very strict plan with adequate safety monitoring in it to advance the compound, because in the end, you're about to put something that's never been put in human beings before into a human being. And the regulators want to make sure that you have a plan to do it well, a trial to do it well. Also includes information about the investigator. The investigator is the doctor responsible for following and implementing the protocol. And then often a new medicine to the United States may be a medicine that's available outside the United States, that's used in other countries. And in those instances, there's a lot of information about the chemical or the protein. You have, there could be clinical trials that have been done outside the United States, but it's never been in the United States before. Even though a medicine's approved elsewhere, it still starts from the same place with an IND, because you can't do any human experimentation or do anything that crosses state lines without an IND. So the part of the IND, this is an important part of the IND for any compound that has been used outside of the US. There is also information on how the drug is manufactured. I mean, think about it. I mean, chemicals are, how they're made, the purity, what they're made of, what the contents are, the ingredients, all critical in understanding safety, especially if you're going to administer that drug to a human being. I mean, even the equipment that's used to manufacture a compound, and then what you're using to assess its stability and purity, all has to be part of this. All the pharmacology and toxicology I mentioned before, and again, that's the information from the animal studies that says what the drug does to the body, which is the toxicity, what the body does to the drug, that's the metabolism, and then your safety assessment I mentioned of the body systems. So All of that is part of it. And it all becomes, this is sort of the building blocks of the IND. And the reason I'm showing this is because this is also the building blocks of the CTD, the Common Technical Document, which is essentially the document that when you get to the end of all of this, which we will get to, gets submitted to the regulatory authorities before an approval. And you're already at this very early stage starting to build that triangle, starting to build up the data that's going to eventually get to approval. And that's, as I mentioned, there's manufacturing information from animal studies, there's information about, there's lots of forms and paperwork, clinical trial protocol. All of these requirements are essentially little pieces and little squares that form this triangle that you will continue to add to throughout the drug development process. And typically at this stage, this IND application is about 6,000 pages. That's about, again, it's a small number relative to the final document, but it is the beginning of your trying to build the story of how this medicine is going to make it to eventually, or I should say chemical is going to make it eventually to be a medicine. So when you submit this IND, what is the FDA looking for? And this is not any different than other regulatory authorities. I think everybody has the same approach to this. First of all, is it reasonably safe to administer this investigational product to patients? The first question they ask. And why reasonably safe? There is very little known at this stage. This has never been in a human being before in many cases. Is it safe based on the animal studies we have? Do we think it's safe at this time to administer to patients? That's paramount to this assessment. And then, are patients being exposed to unnecessary risk? Any medicine, any potential medicine has risk. Are the risks of patients who are about to enter this study, are they being exposed to undue risk? They look at things like the drug quality. Obviously, if you don't have a pure product, that's going to be an unnecessary risk. That's not reasonably safe. Toxicology. Your toxicology studies, your toxicity programs are designed to demonstrate toxicity. They're designed to find those limits of toxicity. However, if those limits of toxicity are too low, it may be too risky to administer to a human. And that's what the animal studies are really looking at. And then the clinical trial. Are the patients being monitored appropriately? Specifically, you've identified these risks from your toxicology studies. Are patients being monitored in case those risks happened in patients? The protocol has to do that. And importantly, because this is the very first time that this compound is being used in a human being, are you starting with the minimal number of patients at any given time to minimize risk? There have been, throughout the history of drug development, very poor outcomes when lots of patients were treated all at once with a new drug. And the outcomes haven't been good. There's been a safety thing and many people got hurt. The plan here is to minimize the number of patients along the way before you have then additional safety information that tells you you can now advance to more patients. And the clinical assessment of your protocol is the way they make that assessment. Now, this is a process that obviously you can see is very complex even at this early stage. So one of the critical ways to make sure that you get off the ground, get on the right track on this is communication. It is very important throughout the process to meet with the FDA and other regulatory authorities before you begin this process. This is even before you are ready to submit that IND. It could be 6 months, a year. You meet with them to talk about what you're doing and understand any potential concerns they might have. They see a lot of things that you don't. You're seeing your program, you're seeing your project, they're seeing the projects of everybody. So they may have seen something else in another program that they're worried about, and any potential concerns that they're thinking about that you might not be aware of, this is where these things will come to the table, and they'll give you advice on how to mitigate some of those concerns. The plan has to be sound, and it has to be strong science. You can't go in to a regulatory authority with a half-baked plan. It has to, all of the elements I've talked about assuring patient safety, minimal exposure, dose finding, all of those things has to be really strong. And it cannot be sort of a theory that somebody might have about that it's going to work here, I know it. No, it has to be rooted in all of those target validation studies from the very beginning that says there's a chance that this compound could be beneficial to somebody. And then, educate FDA, and goes to other regulatory authorities. And this is not at all to sort of say they're not educated. They're actually quite well-versed on the drug development process, but they see lots and lots of drugs. They see lots and lots of diseases. With the way medicine is advancing now, it is impossible that anybody could know everything about every disease and every biological target in a given disease. So you can't assume they have deep expertise in your area. You have deep expertise because it's all you're working on, but they may not. So they also may not know the disease very well. There are a lot of diseases. There's thousands of rare diseases now that are being explored. So part of this process is educating them about the biology, discussing the unmet need, giving them a sense of urgency in terms of how important is it to the community and physicians that this product be moved forward, and extremely important and very relevant in this conversation, patient and caregiver experience. This is a critical part where any patient or their family has the opportunity to give input, and that input is carried by the sponsor, whoever it might be, as part of that education process. This is very important and increasingly important as we again look at rarer and rarer diseases and also less understood diseases. But even in a sense like in a disease like multiple sclerosis, which has been heavily studied and thankfully has treatments, the unmet need in MS is still something that I think there's a lot of unmet need that remains, and educating the agency about the FDA or European regulators or whoever about where that unmet need exists and how patients need us to fill that need is such an important part of the process. So very important part of the communication plan at its core, drug development, but at its core, any good drug development program should revolve around what patients need. So now your IND, hopefully you get clearance. You get no objection. There's no such thing as approval. They say, we don't object for you to proceed. And now you begin your first in human studies. And the first step in that is typically called phase 1. This is your first in human testing where now you have all this animal data, and now you're looking for whether those animal biologic effects that you saw, those effects in the animals, how that now looks the first time you give the medicine to a human being. So the focus of this study is safety and pharmacokinetics. So pharmacokinetics means how the drug moves through the body. Safety is clear. We look to see what is the effect of the drug on the body in terms of toxicities. Pharmacokinetics, how does the drug move through the body? And typically in these studies, we start with low doses and we move very slowly. Again, first time ever given to humans, you could have all the animal data in the world. Humans are different. You just never know what's going to happen. So you start slow and then you gradually move up. And what you do is each dose level, typically what's done is you have an independent group outside of the company, outside of who's running the study, to look at that safety, to get an objective opinion about whether it's safe to proceed to the next level. Because we all have biases, and having an independent group help you in that decision-making is very important. Typically, these studies start with just a single dose and then you observe, but some may have multiple doses. And people will call a phase 1A if it's a single dose study, a phase 1B if it's a multi-dose study. And typically, very small numbers, the minimum needed to answer your question, which is the minimum needed to assess some safety, meaning for more obvious safety effects, or again, how the drug is metabolized. So it's that first step, and again, whether in humans, what you saw in animals is recapitulated in humans. So now you have your phase 1 data, and it looks like, okay, great. We understand more about how the drug is metabolized. We see that based on our human data that we've now collected, the animal data was predictive. So great, we have now human safety data, and we have human pharmacokinetic data. So you will typically file an amendment to your IND or file a new CTA to say, here's our new clinical data. We're ready to move on to the next phase. And that's typically called phase 2. Phase 2 now is when you're trying to determine whether some of your early data on the effect in the disease holds true. So now we have an idea how this medicine or potential medicine, this compound, alters the disease in some way. Phase 2 is often the first time that you're now testing that in a larger number of patients, measuring blood tests, what are often called biomarkers, to assess whether some of those predictions of effect are coming through. Typically, this is also called a proof of concept study. You'll hear that term used. Typically, repeat dosing, usually for a longer period of time. 6 months I give there because in multiple sclerosis, the most common type of phase 2 is like 3 to 6 months. MRI is a type of biomarker. It allows us to understand whether the medicine, when given to people living with MS, when you do MRI imaging of the brain, whether you can change some of the MRI findings. That's a classic proof of concept study. Typically, these are larger number of patients that are, again, to assess safety still, because now you have more patients, but importantly, indicators of efficacy. I gave MRI as an example. You may test 2 to 3 different doses of your medicine. Maybe in phase 1, you found that it's super safe, except you get to the high dose and it's not as safe. So maybe there's 2 middle doses that you think, I don't know which one of those is going to be the best one for efficacy, or for efficacy with the best safety profile. I'm going to test a few of those in my proof of concept study or my phase 2 study. And an important part of this is why we're doing this, a phase 2. Why not jump phase 1 to phase 3? Why we do phase 2 is to determine whether the efficacy and safety in this somewhat larger group than the phase 1, where you're measuring disease-relevant measures, support a larger confirmatory study. And that means support it in terms of safety. So now you've gone to, I don't know, say 50 patients or 100 patients and it looks fine. Now for the next study, which we'll get into, sometimes you need to study a medicine in thousands of patients. Have we now have enough information to help us know if that's going to be safe? Just as importantly, this is an important step where you want to assess your risk of failing on efficacy. I mean, because now this is a study, as I said, 50 to 100 people. You're getting into phase 3 studies with thousands of people. This is the time to decide, have we gotten to the end here and we just don't have a drug? This is the ideal time to say enough is enough. Let's go back to the well and start over. This is where you're assessing your risk of that larger study in terms of safety, as I mentioned, but also efficacy. And what changed drug development in multiple sclerosis is that a biomarker like MRI was realized to be predictive of clinical outcome measures in large studies. So companies could now do smaller studies with smaller number of patients, get an effect, and essentially improve the likelihood of success in those larger studies. Critical step in essentially causing an immense growth in research and development in multiple sclerosis. So this is an important step. Often a lot of time is spent in this step, but it's really critical as a foundation for future studies. So again, biomarkers like MRI are what we look for, and not all diseases have been as fortunate. So very important step in the process, and I think the history of MS demonstrates why. So now you have done your phase 2 and you've seen like, hey, now I got an effect. So I really got something here. I think I really believe that we have a compound that could really alter the course of this disease. You go back to the regulatory authorities. In the US, it's called an end of phase 2 meeting. In Europe, you may go for scientific advice at a centralized review body called the Scientific Advice Working Party. And you share your data. You say, look, I think we have something here. And you solicit feedback on the acceptability of a plan that now gets you from that early biomarker proof of concept data to approval. And you start discussing that because now it's real. Now you're going to make a huge investment in the program. You're going to expose lots of patients. And you're going to expect at the end of all of this that the regulatory authorities are going to find what you did acceptable and then start that process towards approval. This is the time to start having those discussions. You also have to make enough medicine, enough drug to supply the commercial market in case you get approved, because the regulatory authorities and anybody should frown on getting to the end of a trial, showing great effect, and then not being able to produce the drug. Not a good situation to be in. And often at this point, based on your clinical studies, the regulators may ask for additional preclinical studies, they may ask for additional clinical studies, they may ask, as is often very common and required, pediatric studies, things of that nature, again, to fill out that overall approval package. And then you get into phase 3. Phase 3 is your confirmatory. You'll hear it referred to as your pivotal. You'll hear it referred to as your registrational trials. Typically, 2 phase 3 studies are required. Not always been the history in MS. It really depends upon the disease. It depends upon how much the feasibility about whether you can really do 2 phase 3 trials. Depends upon the strength of the data. If you have clear survival data and you have a disease that's invariably fatal, you don't need to show that often in 2 phase 3 studies. But typically, it does require 2 phase 3 studies, typically large numbers of patients. And very importantly, unlike your phase 2, which is about you understanding biological effect in your disease and de-risking your phase 3 program, you need to show an effect on clinically meaningful endpoints. And typically that's referred to as, quote, feels and functions. So things that are truly meaningful to the patient, not interesting from a scientific standpoint, but meaningful to how a patient feels in their everyday life and how you can impact their function. So this is all the beginning, and this is where you get into these studies. And once you have these studies, this forms, again, the foundation of your approval. Now, I took you very linear through clinical trial procedures of a typical scenario. Often, things get combined. So sometimes a company will combine phase 1 and 2, where you're combining safety and efficacy goals. Sometimes you're doing a multi-dose phase 1 and then multi-dose range finding. That's called a 1B2A. Sometimes there's that combination. Sometimes you'll also see enough data from a phase 1 to support a phase 2-3, where your phase 3 isn't really that much bigger than a phase 2, and you have some very exciting information from your phase 1 study, and you don't have to go through this linear process. It's all driven by data, all driven by the science, and all driven by the unmet need and your communication and feedback with the regulatory authorities and the community as a whole, patient and physician community, on what's the best way to get a drug to patients in as expeditious way as possible. So now you have your phase 3 data. Let's say it's positive, very exciting. Things still fail at phase 3. There's no question about it, but at least now you're at the end of phase 3. You have data that you believe supports approval as a medicine, so you submit. You put together an application, and this is called an NDA, or a new drug application. In the US, in the EU, it's called an MAA. Once the application goes into the FDA, they can decide, is it a standard review? So meaning, is it their usual review process, about 12 months, and a priority, or is it a priority review? Now, priority review, which is shorter, about 8 months, a priority review is where the data are addressing and appear to be, at least on the surface, superior to anything out there. There's a high unmet need. Where there's a high unmet need and where the data suggest a major advancement in the treatment of a disease, the FDA will accelerate their process. So whether something is priority versus standard, again, depends on the data, depends on the disease indication. In the EU, the approval takes about 12 to 24 months. There's a Q&A process back and forth during that varies on the time. Now, I mentioned earlier on about this CTD triangle, the Common Technical Document Triangle. And this was something that was created because it used to be that you needed for every country to file a different application. And it was and every country wanted something different and there was no harmonization. So now there's a harmonized process that you put together a single application based on this triangle, with the tip of the triangle, the red part there, the stuff that's really focused on local requirements. So the bottom part, the bottom two segments of the triangle, all of that is required for everybody. And then the upper part, which is the little tiny triangle at the top, that is the part that may have special requirements for a given geography. And this has greatly improved certainly the burden on initiating this process. But it's still a very rigorous process. I've described all of these steps leading up to it. I mentioned earlier how your IND fills in little bits. Now the rest of it, the rest of your clinical program since IND essentially fills in the rest. You add to each of these squares throughout the entire process, and this could be quite a big application. And the last one I did was at over 4 million pages. These are major efforts, and believe it or not, people look at that. They read all of that information. They try, because it's so important that these assessments be thorough, because once a medicine enters the market, it's very hard to handle it if something unpredictable happens. So very robust process that takes, as I think you've seen, many years. This slide just shows your typical review process of an NDA at FDA. This is essentially the timeline that I've just described for you, a little bit more in depth to let you know what's happening at each of these steps. But there's a lot of meetings, a submission, a lot of communication, perhaps additional analyses that continue during this time, again, to arrive at the most robust assessment of the safety and efficacy of the process. Now, this takes a long time. So, how much does all of this cost? There was a recent study that said the median cost of all this is about $985 million. So close to $1 billion throughout the process, with a broad range. 314 to 2.8 is what was cited in this particular article. Neuroscience trials tend to be more towards the expensive end versus other areas. So a pretty big process, a lot of years, a lot of man hours, a lot of patient and investigator commitment to get to a medicine at this point. So in summary, I hope I've been able to communicate that this is a complex process, but what's very important is that there's rigorous testing along the way of any possible new medicine, and that this rigorous testing starts from the earliest time of coming up with an idea all the way up through human studies, and then a very rigorous review process. It's a heavily regulated process. Patient safety is at its core, but also benefit is an important part of the equation. Good communication at each stage of development helps minimize the risks along the way, helps facilitate this to move things forward faster. And even though often, especially in an area like MS, there's all a history of how drugs have been developed in MS. And people often get comfortable, well, we'll just do what everybody else did because that's low risk. That's the wrong way, in my view, to develop drugs. You have to continually look for innovative solutions to lower the risk of failure and accelerate pace while minimizing risk to patients. I'd say very important still to this day in multiple sclerosis. And this communication and this process of developing those innovative solutions and trying to advance things in a fast but safe way, patient input is so critical. I can't emphasize that enough about how important it is that companies and regulatory authorities hear from the community on what they need and how they would like to see medicines, how they would like to benefit from medicines. And it's this communication process, and this is where the influence can happen at this level of these innovative solutions, because there's still a lot of challenges in multiple sclerosis waiting to be addressed. So with that, again, thank you for your time. I really appreciate being invited to give this presentation.
Thank you, Dr. Panzara, and thank you everyone for watching. For more information, please visit cando-ms.org. Thank you very much.